and programs, and Hillary Penner is right over there. Yeah, she's a director of program development for education, and both of them don't live here in the Keys, but we work very closely on a daily basis. So it's really nice to have them here with us in person, and we thank them for all of their support. Would you like to say a word, Martha, or no? Okay, that's fine. I know. We're eager to get going here. So then without any further ado, I will turn it over to Lindsay and Ellery for their presentation. I'll let you all introduce yourselves, anything you think we need to know about you. And yeah, we're ready to hear about it. Thank you.
I think I turned the mic on. Okay. I am Lindsay, and Ellery is lurking over in the corner for the moment. And we're going to sort of tag team this talk today, telling you guys about the use of videos for fish surveys. We both work at the South Florida Regional Lab of FWC, so we are just an hour down the road in Marathon. Yeah, so as I said, I'm Lindsay. A little background on me. I got my bachelor's in biology at Elon University up in North Carolina, and then got my PhD at the University of Florida. Thank you. And now, as I said, I work for FWC. I'm part of the restoration ecology team. And I focus more on the coral side of restoration, although our team does focus on a lot more, which Ellery will tell you about. And my projects range from coral nursery optimization to the husbandry and grow out of the long-spine sea urchin, Diadema antillarum, and then putting both the corals and the urchins back out on the reef. So I'm going to pass it over to Ellery, who will tell you a little bit about herself and start off this presentation.
Switching spots. I gotta look at Diadema. Perfect. So I'm Ellery. I did both my bachelor's and my PhD at University of Miami, go Canes. And I also work with Lindsay at FWC, down on the restoration ecology team, except I focus more on nearshore hardbottom habitats. And some of my projects range from optimizing the sponge nursery to some new and upcoming octocoral restoration trials. So we're here for Fish and Friends. We're going to talk about video surveys, but we're going to back up first and talk about why survey fish. We really want to understand their roles in different ecosystems and on the reef, and also see if there's maybe a lot of redundancy between different fish species and their roles. We can also use them as an indicator for the overall health of an ecosystem. We can look at fish, how they change over time, maybe before and after an acute event like a hurricane, or just over the span of decades to see how our systems in the Keys have changed over time, as well as over space, maybe close to and further away from a construction event or a new marina, to see how our ecosystems are changing. Similarly, we can also use fish to gauge the success of restoration. Some of the work I do with our sponges is outplanting them. Now, we would hope that that would attract more fish, that we'd see some of these positive effects cascade up the trophic web. So we have to study fish to figure that out. And different conservation efforts, such as closures and protected areas, are typically designed to protect fish. How are you going to see if they work if you don't survey those fish? Finally, we of course depend on lots of fishery species, so we want to monitor those populations to make sure that we are not over harvesting, to ensure that we have healthy and sustainable fisheries long into the future. So I think I've convinced you it's important to actually survey fish if there was any doubt before. And now, how do we survey fish? The most common fisheries independent surveys all kind of fall under this large umbrella category or term, underwater visual census. And really all that means is any survey that requires, eyes in the water, whether that's a diver or a video camera. These are non-extractive surveys, so you don't actually have to kill or remove the fish to survey them. So they're much less destructive than maybe some of the old types of methods like cyanide fishing, and much less disruptive than, say, like seining or bottom trawls. And so they might be required in certain management areas. And again, there's two kind of main types of these underwater visual censuses, diver-based or video-based. So starting off, talking a little bit about these diver-based surveys, another broad umbrella term. There's tons of different types of these diver-based surveys. REEF does some of them. And this isn't the focus of the talk, but I did want to touch on them because they're so common and popular, and they are kind of like the standard that we hold newer methods like video surveys too. So some of the benefits of actually having a diver in the water, oftentimes they're mobile, they're moving around, so they can survey different microhabitats in a larger area, and that allows them to include species with patchy distributions as well as maybe more sedentary or even sometimes cryptobenthic fish that are kind of hard to see. Having a diver in the water recording abundance, if you're also doing it in a known area, you can then use that to calculate density, which is a really useful metric for fish. And it's usually pretty easy to train a diver or snorkeler to estimate fish length. Then you can calculate length distributions and for certain species as well as biomass. Some of the most common types of diver surveys are transects, roving diver surveys, which a lot of those reef citizen science fall under roving diver surveys, and then stationary point counts. And because they're so common and well accepted, that really promotes sharing between different researchers, different institutions, and even like over decades. I can look at data from the 80s and 90s. It's going to almost certainly be one of those three types, really easy to replicate or look for other sources and get comparable data. Some of the negatives, just having a diver in the water does affect the fish behavior. Many fish will try to avoid the diver, thus leaving the survey area and not getting counted. Some fish are really curious and will actually come into the diver's space to see what they're doing. Either way, there's a bias there. But overall, typically we do see fewer fishes being observed when there is a diver in the water. And it's hard to believe, but all people, including divers, do sometimes make mistakes, whether it's misidentifying or miscounting. And then, of course, there's possible safety concerns, whether that's depth, exposure, temperature, boat traffic, or just like the duration you want that survey to happen at a certain depth, which then brings us to our video-based, so the other kind of main umbrella term for different visual censuses. And some of the main pros here is that there is no diver effect most of the time. It also does create a permanent record. You have to take these videos, watch them on the couch at home. You can pause it, get up, grab your fish ID guide if you're unsure of a fish, so you maybe are getting more reliable identifications. They're also easy to replicate, and generally speaking, they record similar fish species, both transects and these, or both any diver-based survey and video-based surveys are going to get the really common and abundant fish in different systems. There's, of course, still different biases there with what they're better at getting. And they're really good if conditions are not suitable for divers. Whenever we think of depth, we think about it being too deep. But also, you still want to survey fish in really shallow waters. And sometimes it can be really hard if there's a silty sediment type to get divers in the water without just absolutely ruining the visibility. And in this figure, you can kind of see this is like a bird's eye view looking down at what a stationary video camera might capture. So you do get a limited field of view. A lot of times they are stationary, they can't move around, they could potentially miss fish that are hiding behind a large coral head. And the way you count fish in these videos, it's the maximum fish in a video frame at any given moment. So we can't actually use that to calculate density, but we get a relative abundance index. And then finally, it does require video processing back on land. So really nice to be able to verify that ID, but that does extend the time it takes to actually get useful data from these surveys. And also I want to be clear, I am not advocating for one type or another here. They both have their pros and cons, inherent biases in the types of fish that they can attract or document, but they're both great options. I'm not advocating, but we're just here to talk about videos today. The best method for any one person is going to depend on the environment and the actual research in question or species you're interested in. So going on to some types of video surveys, we can have them baited or unbaited. Unbaited, you're getting a more general assessment of the fish in that area. But then you can also bait those videos to try to target specific species if you're interested in certain groups. Typically, we think of some chum to attract large predatory fish like our ferocious bonnethead shark. But this has also been expanded. So the University of Miami is actually using coral as bait to attract fish to try to see what fish are munching on their new baby coral outplants to try to work on mitigating that. And one of the projects Lindsay's going to talk about, she used algae as bait to look at feeding preferences. So it doesn't just have to be chum. There's also, you can get, most video surveys have a single camera, but you can use 2 cameras for a stereo video system. And then you can use photogrammetry to estimate fish size. So that does let you estimate lengths and stuff that normally you'd need a diver in the water, but this requires math. Most videos are stationary. You just kind of stick a camera on a weight or leave it out there. And so you're only going to record the fish that actually swim past the camera, but you don't get that diver effect. You can also, though, use mobile videos, whether it's a remote operated video or diver operated. But now you're kind of getting back into that diver effect, but that also might be more comparable to transects if you have that video kind of moving over a transect tape. So now that y'all know a little bit more about types of video surveys, etc., we're going to talk about how we actually use them for fish research. So we'll see how they are used for characterizing fish communities in a few different systems, how they can be used to study behavior, which again, you really kind of don't want people impacting their behavior if that's what you're interested in, as well as examining population trends for species of fisheries interest. So this is actually some of my dissertation work from So this is all up in Biscayne Bay in Miami. And originally I was looking and comparing transects to video surveys in the mangroves. And then I also used those video surveys to characterize the fish communities. So I looked at a couple of sites. Chapman is this northernmost red site. It's adjacent to a neighborhood with a bunch of residential canals. Still has a good bit of mangroves, and it's characterized by having a higher salinity. The southernmost site is by the Deering Estate, which has some seawalls and like kind of a dredged canal area, and it's characterized by having a lower salinity. So I expected to find different fish assemblages between these two salinity regimes. I did not. The fish communities were the same between those two altered mangrove shorelines. Did that? Oh, it does work to click. So this is just an example of some of like the really high diversity we would see in the mangroves around Miami. We've got sergeant majors. Those are some striped mohara, a gray snapper. There were a bunch of rainbow parrotfish that moved through, some sailor's choice. We've got a schoolmaster snapper with those yellow fins there. But then, so you see all the diversity we get with the camera. And most of the videos weren't quite this busy, but this was a 1-hour video and it took me 10 hours to process it, like on the couch. So you really do get to kind of see the pros and cons of the videos here. And then actually looking at some of the community characteristics we saw, I was interested in looking at them by their different feeding groups. And the fish in these mangroves were absolutely dominated by invertivores and piscivores. And there's a lot of flexibility between invertivores and piscivores. Fish typically aren't picky. They're going to eat what's an appropriate size for them to eat, and their diets will shift as they get older. So pretty much any of these piscivores will also eat invertebrates, and most of these invertivores will also eat fish. There were some planktivores. Those were typically represented by those small water column, those schooling silvery fishes like silver sides and herrings. Detritivores are represented by the seasonal mullet run. And then we had parrotfish and dancerfish occasionally, which were our herbivores, but really mostly invertivores and piscivores. And just for your viewing pleasure, some videos to also really kind of show the mobile predators that we were able to get in our videos. We had a little baby lemon shark moving right through there. The mangroves are green. It's kind of hard to see too clearly in the water, as well as more baby lemon sharks. We froze. I was comparing these videos to transects, so we were able to see predators in the videos that the transects didn't capture, including the lemon sharks, as well as bonnet head sharks. There he goes. Okay, so that's, I swear there was one in here earlier. We were also able to capture jacks and drums, which weren't seen on those transects. So that kind of gives you an idea of the different types of fish you can capture with videos that you don't get on transects. But then, of course, you're not getting the small or sedentary fish that don't swim in front of your camera. So there was one more video that I thought was really significant, but I wanted to explain why it was significant before I show it to y'all. So most people in South Florida are kind of aware of all of the alterations we have had to our hydrology. So over 100 years ago, the water from like Lake Okeechobee was diverted away from the Everglades. So Biscayne Bay and Florida Bay were historically much more estuarine than they are today. Now they're mostly, a marine salinity regime with marine fish communities. One of my sites, the Deering Estate Flowway, was part of the Cutler Slough rehydration project of the Comprehensive Everglades Restoration Plan. So construction on that site was finished in 2012. So the water flow has been restored. They're trying to best mimic the historical freshwater flow with seasonal pulses from Cutler Creek into Biscayne Bay via mangrove creeks. Here we go. No, too far. We're just going to click on it this time. Cool. And I was able to capture a pretty sizable black drum. So before the hydrology was altered and Biscayne Bay was estuarine, black drum were reportedly very common. And they were one of the species that were lost when the community shifted from estuarine to marine. So, and also I keep mentioning I was comparing these to transects. NOAA has conducted 25 years of transect surveys at about 50 sites twice a year. That's a lot of math and a lot of transects. They did not get a single black drum in any of those transects. And I was able to catch one on the video, and whether that's actually due to the biases of the methods or just because they're that rare, that was really cool to get to see this estuarine fish. And one fish observed one time is absolutely not a metric of success for this restoration, but it gives me a little optimism. And knowing that they are in fact there was really exciting to get to see that, this rehydration restoration project might be attracting estuarine fish back to these the Deering Estate Flowway. We're done with the mangroves. So now we'll be talking about characterized communities on artificial reefs, and now we're back to these. So there we go. We're doing some research at the Long Key Artificial Reef, and these were created in the 80s using the rubble from the old Long Key Bridge. So you can see all of these dots in different arrays. Each of those is a little artificial reef. And again, these were put out in the 80s, so studying on these artificial reefs gives us an idea of the climax communities that we can find and gives us an idea of what to expect if we are going to put out more artificial reefs in the future. So we've been surveying the fish communities, actually, But for the flow of the talk, it made sense for me to fit it into the first half. So we've been surveying 2 reefs within that whole array every single month using video surveys. So you can immediately see a lot of fish and a lot of diversity. We have tomtates, there's a 4-eye butterfly fish, a blue striped grunt, hogfish, some parrotfish. And some of these fish are coming right up to the camera. They don't really come up to divers like that. Okay, now we've got some wrasse at the bottom, rainbow parrotfish, another hogfish. Graysby, muttons, and you kind of get a much better look, again, just without a diver. We've got a white grunt, some more bluehead wrasse. But yeah, sometimes I'm like, are they actually coming up to check out the camera? Because they don't come up to divers like that. And sometimes I'm kind of glad the barracuda don't come that close to me when I'm in the water. I know they're fine, but they're still pretty toothy. Maybe a little bit. I do wish the spotted trunkfish would come up that close to me when I was in the water. Just want to give them a little kiss. And then the obligatory charismatic megafauna reel. So southern stingray. What's the depth here? 30. A nice turtle. We also started aiming the cameras a little further up. We're just getting a bunch of bellies. But speaking of bellies, we got some bull sharks, and at least with a belly view, we could see that most of the bull sharks in this area are males. And we were getting more of those bull sharks. We would sometimes see divers in the water, but we tended to detect more using the videos. And they didn't swim away from the videos like they swam away from me when I was like, ooh, shark, and chased them. So this is still an ongoing project. I haven't even finished getting through all of these videos. So this is a very, rudimentary look at the fish communities here. And once we get a full year, we'll be able to actually detect seasonal differences as well, potentially. But you can also see, just like the video showed, there's a lot of diversity. Some of the main families are the wrasse, grunt, snapper, parrotfish. And with that, I'm going to turn the microphone off and pass it over to Lindsay to talk about fish communities at solution holes.
Okay, I'm going to pull everyone a little closer to shore again and talk about solution holes. This is actually an ongoing project the lobster team down in Marathon is working on. And solution holes are very unique. They're essentially these mini limestone caves in the middle of seagrass beds and sandy flats and other nearshore habitats. And having these little caves, these little shelters in the middle of what can be a fairly barren seascape really acts as shelter, and all these different fish and invertebrates will gather in these solution holes and utilize them often in a juvenile stage before moving off to the reef. But with these solution holes and them not being the most understood ecosystem, they're fairly unique. Lobster team set up cameras because, as previously mentioned, cameras are a great way to really catch what's going on when no one's around. And cameras were also able to be set out for a 2-week period where they could continuously monitor who is using these solution holes. And okay, there we go. And you'll see, again, we've gotten near shore, so we're very green again, but can see a whole diversity of different fish species that you might not expect to see. Like if you said, okay, we're gonna go out to the seagrass, you wouldn't expect to see, yeah, we can't avoid all the cute little trunkfish and things. But we have a moray eel, we have a hogfish, we'll have some grunts come through. Like there's a whole variety of different fish that might use these habitats. And of course, that adorable little friend. He does. He does. The other very unique thing about solution holes is the role of grouper with solution holes. These grouper actually act as ecosystem engineers and will keep the solution holes clear of sand so that they and other fish, I mean, they do it for themselves, but other fish can also utilize these solution holes. So you'll see our friend here just hanging out outside a solution hole. He'll go in, again, same solution hole, really, this is where he wants to be, and he will make sure that he gets to keep his little area. This is his solution hole, and no one is allowed in it. So, no fish were harmed in the making of this video, I swear. But they are, it's his solution hole. Like, he wants to keep it intact, he wants to live there, and benefits most fish. Sometimes he bullies away some bigger guys. So along with looking at the role of fish for habitat modification, I focus for my PhD in looking at feeding preference. My PhD topic was the role of herbivores in shaping benthic communities, and a big part of that is algae. And part of that is understanding which kind of herbivores are going to eat which kind of algae. Because not all algae are created equal, and not all herbivores are created equal. So knowing what might be on a reef if you have a lot of parrotfish, or a lot of sturgeonfish, or hopefully a lot of diadema is very important. But although you can put a diadema in a tank and it will behave pretty naturally, fish won't do the same. So I had to use videos. So it's a fairly simple method where you just can put known algae on a line, attach it to the bottom, let those lines float up in the water column, and any fish swimming by can pick and choose. It's a ready-made buffet for the fish in the area. Sometimes you have to move a line because damselfish are jerks and will bite anything they can. So this was basically what I spent hours of my life looking at. Is we'd have, again, a 1-hour video with my known algae. In this case, we had a structurally tough brown algae, which was my turbinaria, a chemically rich algae Laurencia, seagrass, thalassia, and then halimeda, which is a very crunchy calcareous. And so I wanted to know, what do these fish like? So you can see here, I thought maybe this parrotfish would go for it. No, he decided not. But our surgeonfish here really went after the Laurencia. And so to look at feeding preference, I'd count every single time a fish took a bite of an algae, which is fine for the parrotfish who take these nice big bites like that. But the surgeonfish will take 100 little bites all at once, and then you have to try to count everyone. So fun times with videos. Sometimes we also got unexpected things on these videos. I was looking at feeding preferences of herbivores, but I got a little carnivore action. So if you watch our little yellow fish at the top, likely a juvenile bluehead wrasse, he will be minding his business and then narrowly escape predation by our little grouper friend. So, I might have wanted to know about herbivores, but I got to learn a little bit about some carnivores too. Yeah, he escaped. The grouper had to try again. So this is just some basic results about the different feeding preferences. So I set up these assays both at a shallower reef crest around 5 to 10 feet, and then a deeper spur and groove reef around 60 feet. and looked at how the different fish, what they wanted to eat at these two different sites, and if that might vary. You'll see we have some non-herbivores, like the blue-striped grunt, who decided to try out seagrass and then just swam away because it's not an herbivore. Our chub also did the same. But then we have our surgeonfish and our parrotfish. You'll notice things like the stoplight parrotfish really had a favorite. He really wanted to eat seagrass, while our surgeonfish and our yellowtail parrotfish kind of tried a little bit of everything, although certainly did have a preference. And you can see the same thing at the deeper reef, where the surgeonfish and the parrotfish might have tried a little bit of everything, but had preferences. But interestingly, their preferences were different, shallow and deep. So this does tell you, like, these different fish communities affect the algae on the reef differently. So that was a lot of my PhD. In case anyone's interested, urchins and fish don't like to eat the same things, so you need all of them. Next, I'm going to be talking about a big project that our finfish team has been working on, and this is Western Dry Rocks. Western Dry Rocks is a site about 10 miles southwest of Key West. And this site is important because it is a spawning aggregation. It's been found that both snappers and jacks gather in this area to spawn. And due to that, based on that information, there has been a seasonal closure there since 2021. Between the beginning of April and the end of July, it's closed to all fishing. So this is obviously very interesting, and we want to know more, but these sites are deep and far away. So again, we can use videos to find out information we just wouldn't be able to otherwise. So we can use videos, and if you're lucky, you get a nice cool video like this of a bunch of permits swimming conveniently right in front of the camera for us. And with this, we can see not only the fish community in the area, but any spawning behavior that might occur. And as I said, you can see what's around, so we can understand some population trends. This project has 4 main target species, which are gray snapper, mutton snapper, permit, and yellowtail snapper. And with the use of these videos, we can answer 3 main questions, which are: what target species, which of these target species are present in Western Dry Rocks during the closure? When is the activity of each of these species highest and lowest during the closure period? And how do the relative numbers of these target species change from year to year? There's been a closure since 2021, so how was the population different in 2021 versus now, several years later? And along with us, as Ellery put it, sitting on our couch and watching fish videos, we also have started using some citizen science. Specifically, we're using a website called Zooniverse, in which people all over the country can upload their projects, and anyone, anywhere, can look at these projects, find something they're interested in, and help out with some of the science. So this is not just FWC, of course. There's a whole variety of different projects that people can take part in, ranging from, of course, corals and fish, to birds and snakes, and even you can take part in looking at old shipping records and trying to find shipwrecks and where they might have ended up in the 1700s, sort of anything you might be interested in. So we're using Zooniverse for both our Western Dry Rocks and our Solution Holes projects. Well, we will be soon. Will be called Marine Lens Florida Keys, and it's about a month away from being launched on Zooniverse. It would have been great if I could have pitched that right now, but with patience, anyone who wants to help out certainly can. And with that, you will be able to, at the top is an example of Western Dry Rocks videos you might watch. This is actually that permit video. again, and you can count the number of permit you see and note, are there any spawning behaviors or color changes that also indicate spawning? Same with our solution holes work, where you can note what species show up on these videos and any behavioral changes. So I know we dumped a lot of information all at once. So if anyone is interested in learning about these projects or any other projects that FWC is working on, there are some links here. Essentially, everything got moved. If you look up FWC research on most social media, it is there. Also, because I teased you with Zooniverse and then said you couldn't help out, I was going to tell you about a Zooniverse project that is online, and that is our Everglades Wildlife Watch. This is a FWC project in which you can look at stills and videos from the Everglades and help identify the different organisms you might see. And I've looked through it a little. There's some really ridiculous pictures of like deer and stuff doing things like sticking out their tongue. It's very cute. So I want to thank you guys for attending our talk. And are there any questions?
Where and how did you harvest the seagrasses or the macroalgae that you were using for your strings?
Where did I get my macroalgae for my lines? So that project was not done in the Keys. It was actually done in the Cayman Islands. I was lucky enough to go down there for my graduate work. And I first did a survey all around the island to identify the most common macroalgae, and then collected those, with the exception of the seagrass, which is a positive control. I know that fish like it, especially the parrotfish, so that was not unexpected results.
And then my second question, are you finding a lot of stone crabs in your solution holes, or are these like old stone crab homes, or these are built into the reef in other ways? Like, you're not finding stone crabs?
There are some stone crabs, I'm sure. This is very early on. They just did these videos this summer, so it's still a lot of going through the different videos. But, you know, lobsters certainly